Welcome to BGI's first webinar on the topic of decoding the deadly outbreak strand of E.coli. My name is Joyce Peng. I'm the Marketing Director of BGI Americas. Today, we also joining us, we have two distinguished speakers. The first one is Dr. Liang, and she is a postdoc scholar from Technical University of Denmark, and she was a visiting scholar at the University Medical Center at Hamburg in Germany. She played a pivotal role in tracing the pathogens of the E. coli outbreak by genome sequencing, and her research focuses on bacterial pathogenesis and microbial evolution. She has published 23 scientific publications with more than 500 citations in Google Scholar. After that, we will have Dr. Peter Chin Hong, and he is an associate professor and also a clinician in UCSF. And he will share with us a clinical view of this topic and talk about its relevance to people in the United States. After that, I will share the results from the work we did at BGI on sequencing and analyzing the outbreak strand and how we developed the diagnostic protocols which we made publicly available. In the end, we will have a Q&A session where you can ask questions to Dr. Yang and Dr. Chin Hong. In addition to that, we also have Dongfang Li from BGI, who is the lead of the bioinformatics analysis work for this project. She's joining us from China to answer your questions as well. Okay, without further ado, here is Dr. Yang.
Hello, my name is Liang. I am a postdoctor at the Technical University of Denmark. I was working as a visiting scholar in Department of Microbiology at the University Medical Center Hamburg Eppendorf this summer. So we have experienced the EHEC outbreak, and I'm going to tell you some detail of this outbreak. Okay, now we will have an overview of this 2011 E. coli outbreak in Germany. This figure is borrowed from the Robert Koch Institute, which is the CDC of Germany. It shows the timeline of the 2011 E. coli 0104 outbreak. The light gray bar indicates how many infected cases were reported per day. As you can see, in May 22nd, the reported case reached a number more than 200. On this day, the German health authority said, clearly, we are faced with an unusual situation. And on May 26th, the German health official announced that cucumbers from Spain were identified as the source of E. coli outbreak in Germany. And then the EU, the European Commission, on May 27th said that 2 Spanish greenhouses that were suspected to be the source had been closed for investigation. And the funny thing is, the cucumber sample from the Spanish greenhouse did not show any E. coli contamination. And nevertheless, the Robert Koch Institute advised people don't eat raw tomatoes, cucumbers in Germany to prevent further case. However, soon, in May 31st, the lab test showed that 2 of the 4 cucumbers examined did contain toxin-producing E. coli strains, and most likely because of cross-contamination in Germany, according to the expert, but not the outbreak 0104 strain that was found in the patient. And this result indicates that the previous announcement of the Spanish cucumber is not right, but this has already caused a dramatic economic damage to all the farmers. And according to the report, EU farmers claim to have lost up to more than $600 million during this crisis, and the EU finally offered $300 million compensation to the farmers for their loss caused by the outbreak. And this is such a pity. The German expert could do a better job to avoid this, and let me explain why the authority will announce that cucumber is the cause of the outbreak in the beginning. And this slide shows the traditional diagnosis protocol of EHEC from the Institute of Hygiene of University of Münster. As you can see, the whole diagnosis protocol is based on the characters of the E.coli O157 strain, which is the most common strain of EHEC outbreak. Many of the laboratories and hospitals in Germany have equipped with the diagnostic kit for this O157 strain. So during the outbreak, this diagnostic kit will of course be used to screen different samples for EHEC strains. And they have found STX positive E. coli strains from cucumber. This is why they announced that cucumber is the cause of this outbreak. And actually, later genome sequencing analysis showed that the 2011 outbreak E. coli O104 strain does not contain the full STX gene at all. So if the German experts start the genome sequencing 1 week before, they may not give the false announcement saying the cucumber is the cause. So when the German scientists start to think about genome sequencing, showing here in this slide, it is actually quite late, and that's after I came back from Copenhagen to Hamburg after a short visit. So I arrived Hamburg in May 22nd, and then on May 23rd is Monday, my supervisor, who is a main diagnostic medical doctor in Department of Microbiology at the Medical Center of Hamburg, told me the serious situation of the EHEC outbreak. Then I immediately proposed the idea to send the DNA sample of the outbreak strain to BGI for genome sequencing. I know actually there are a lot of genome sequencing facilities in Germany and even in Hamburg. However, I think this is a rather emergent case, and we need extremely fast sequencing and want to get the result as soon as possible. And I know people from China work overnight for this. And actually, we are very glad this is the case. After receiving the sample on May 27th by FedEx, BGI used the latest generation of sequencing facility and continuously worked for several days without rest. And on June 2nd, BGI already provided the draft genome data and uploaded it to a different freely accessible database to share with the public. Then several groups immediately started to design new diagnostic kit based on the new genome. So now the outbreak is over. We are planning to do some further analysis of this EHEC outbreak strain. For example, we would like to develop some gut infection model of the E. coli O104 H4 strain, and we would like to identify some virus factor of this strain by transcriptomics and proteomics analysis. So what we have learned from this outbreak, I think the most important thing to do during the outbreak now is to get the genome of the outbreak organism. And we have a Chinese old saying, knowing your enemy and yourself, then you can fight 1,000 battles without the disaster.
And our next speaker is Peter Cheng Hong.
So what I'm going to do is to put this epidemic into the patient context. My name is Peter Cheng Hong. I'm one of the faculty in infectious diseases at the University of California in San Francisco in the United States. So I'm going to present two quick cases to illustrate some features of this epidemic on the clinical level. Case one is a 5-year-old male who presents with 1-week history of bloody diarrhea and vomiting. On admission to the hospital, he has no fevers. His heart rate is 121, which is high. He's pale. He has anemia with a hematocrit of 21. He has low platelets and a high creatinine, showing that his kidneys aren't working well. He requires hemodialysis and multiple blood transfusion. And his younger sister also develops bloody diarrhea 4 days later, also requiring hemodialysis. So this is a typical presentation of hemolytic uremic syndrome associated with E. coli 0157 or some of the sugar toxin producing strains of E. coli. Case 2 is a 40-year-old female presenting with a 3-day history of bloody diarrhea and abdominal pain. Like the kid, her hematocrit is 30, so it's low. She has low platelets and a high creatinine, showing that her kidneys are not working well. She also requires hemodialysis and multiple blood transfusions. This is a new phase of the sugar toxin, E. coli, outbreak as what was seen in Germany earlier this year. E. coli is seen on an agar plate here with this green sheen. This is an EMB plate. So today in these few minutes, what I'm going to do is talk about the epidemiology of sugar toxin produced in E. coli, which is a name that is commonly used here in the United States to refer to these enterohemorrhagic strains, or EHEC. We're going to understand the clinical presentation and consequences of infection with sugar toxin producing strains of E. coli. And throughout all this, we will talk about the role of politics and society in food safety. What is E. coli? It's a commensal found in large bowel of most mammals, and certain strains may cause disease. Many of these E. coli exist in harmony with the body, but some of the strains can cause disease, such as urinary tract infections, meningitis, and diarrhea. There are many kinds of E. coli, and the one that we're focusing on today is the sugar toxin producing E. coli strains, or STEC. This is also known, as Dr. Liang pointed out, as enterohemorrhagic E. coli, or EHEC. The O and H refers to the somatic and the flagella antigens, and they're used to fingerprint each strain of E. coli that is an STEC. The ones that we typically think of as causing hemorrhagic disease leading to HUS, or hemolytic uremic syndrome, are usually the 0157 strains. But what we've seen in the last few years is an emergence of the non-0157 strains, such as what was seen in this epidemic, the 0104H4 strain. What are sugar toxin producing E. coli, and how are they defined? Well, it's a group defined by strains that produce sugar toxin. Sugar toxin gets into the bloodstream, and they are receptors for sugar toxin on the epithelium of kidney cells, for example. This causes damage to the epithelium, and when blood vessels pass through the blood vessels that are damaged, they get, the blood itself gets misshapen, and this leads to anemia. You don't need very much sugar toxin producing E. coli to cause disease, and you can see on the slide that you just need 10 organisms, and this is in contrast, for example, to Salmonella, where you need 10 to the 5 organisms, and to Vibrio cholera, where you need 10 to the 5 organisms. How do you get STEC, or sugar toxin producing E. coli? Well, most of it is foodborne, so meat, poultry, fruits, juices, and greens. In fact, 2 to 4% of store-bought meat is sugar toxin producing E. coli positive. There's been legislation in the United States since the 1993 outbreak that some of you may have heard about in Jack in the Box, which left 4 people dead. And in 1994, the USDA mandated testing of STEC 0157 in ground beef. But as I had mentioned, although there's been legislation that aims to identify the 0157 strains seen here in blue, these tests are not identifying the non-0157 strains. And you can see in the red that there's been a gradual increase in the number of cases in patients due to non-0157 strains. In the German outbreak, there are several features that were different from the features previously seen with E. coli STEC types. And you can see in the dotted line, most of the outbreak in previous years has been seen in younger individuals, like case 1 that I presented. In the German outbreak, seen here in the blue lines, most of the cases occurred in adults. They were mainly in females compared to males, with a median age in the 30s. And Joyce and Dr. Peng is going to talk about this later on, but the current strain seen in the German outbreak of sugar toxin producing E. coli has features both of that are characteristic of a strain that produces sugar toxin, but also features of another kind of E. coli, allowing the E. coli to stack like a group of bricks along the epithelium. In terms of treatment, our treatment recommendations in the hospital is really supportive. There's some controversy, but typically we do not recommend giving antibiotics, and this is based on some previous data showing that when you give antibiotics, you have a higher risk of developing kidney complications compared to individuals who do not receive antibiotics if you have this kind of E. coli. So for now, we avoid antibiotics. How do you prevent it? Some people are working on vaccines for livestock against E. coli, but ultimately the best kind of prevention may be legislation. And earlier this year, there was a modernization to the Food Safety Act enacted by President Obama. Thanks a lot for your attention.
Thanks, Peter, for the excellent introduction. Now I'm going to share with you some of the work we did at BGI in decoding the E. coli genome. So this graph summarized the discovery timeline. And you will see we first received the sample from Germany on May 28th. And then we did the sequencing within 3 days using the ion torrent third generation sequencers. And then we released the data and then developed the diagnostic kit PCR based within a few days. And then also in the second generation, at the same time, we used Illumina HiSeq to sequence to correct some errors and then to give us a near complete map. And then we did a PCR validation and using the Sanger sequencing and then to help us close the gaps. So we employed multiple technologies. So here I'm going to talk about a little bit about their pros and cons and why we chose them. So the ion torrent, we used it's the shortest turnaround time. So that's why we could generate the results so fast and use that to do the PCR primers that was used in the clinic. However, it's lower throughput and it's less accurate. So we use the HiSeq, which has high throughput and then more accuracy, although it takes longer, so the results takes longer. And then the first generation Sanger was used because it's accurate, and then for the for the complete map. Okay, and then so in more detail, the initial sequencing was done from a DNA sample, which we call it TY2482. It's from a 16-year-old girl patient received from the University Medical Center in Hamburg-Eppendorf. And then we used the ion torrent PGM platform for doing that. And then the major finding was that the genome size was so the genome size was about 5.2 megabytes, and then the serotype is 0104, and then the strand has about 99.8% sequence similarity with EAEC 55989 E. coli strand. It's from an HIV patient from Central Africa, and we also identify 8 groups of antibiotic resistance genes. And so we will look at the results. So then we used the HiSeq 2000 to generate a complete map and then identify the causing genes. And then also we conducted more thorough phylogenetic analysis and suggest that the strand is the EAEC strand, which acquired Shiga toxin genes. So here I'm showing you a map. I'm showing you a map, and then you will see Here, this is the final draft of the genome. It shows the disease strand has a circular chromosome of 5,278 kb in length and 3 additional plasmids of 89 kb, 76 kb, and 1.5 kb in size. The chromosome contains 5,000 predicted coding sequences covering 87% of the genome. The bigger plasmid here is highly homologous to a previously sequenced plasmid isolates from a horse and carries additional multi-drug resistant genes. The smaller plasmid carries only 2 genes, one of which encodes a DNA replication protein. The other carries the aggregative adherence fimbria, AAFI gene cluster, which is associated with E. coli aggregation ability and virulence. BGI researchers found that the Shiga toxin coding genes were likely encoded by a viral prophage that integrated into the bacterial chromosome. We also identified several insertion hotspots, including a nested multi-antibiotic resistant associated locus. This indicates that horizontal gene transfer events may have played an important role in the evolution of virulence and drug resistance of this strand. So the availability of the genome sequence enables us to trace the history of the bacteria. The latest analysis indicates that the current outbreak strand has the same profile as this 01095991 strand, which was originally isolated in Germany in 2001. And it's the same in all 12 virulence genes and 7 housekeeping genes listed here. However, at some point over the last 10 years, the new outbreak strand seems to have developed the ability to resist many additional types of antibiotics. And the other strand listed here, 55989, that is similar in sequence, is isolated from an HIV patient from Central Africa, which we just mentioned. Although this Africa strand is genetically more distant because of the Shiga toxin producing genes, and also the telluride resistant genes were both absent there. Yeah, so but these two are the close strands. Unfortunately, the 2001 German strand currently has no public available genome sequence. So therefore, the closest strand that with the sequence available is 55989, and that's what we use to do additional analysis. So in here, we are showing you the results that we compare the outbreak strand with genomes of 90 sequenced E. coli strands downloaded from online databases. This includes 30 finished genomes and 60 draft genomes. And here we are showing the phylogram generated from the SNP data of all these 90 strands by a neighbor joining approach. And this demonstrated that our strand is closest to E. coli 55989. We also conducted phylogenetic analysis using the complete sequence that's available. And from here, it also shows that the outbreak strand is have very close relationship to this 55989 from Africa, with an average nucleotide identity of 99.8%. And it clearly shows that it's the type of EAEC. So we did further analysis of this strand, and our sequence comparison showing that this prophage is very similar to this phage gene. And so it's probably originated from there. And here is a list of these 8 different types of antibiotics resistant genes identified. The first 6 is in a composite transposome in the main chromosome, and you'll see that you also see them showing up in low, although with low frequency, in other E. coli strands. And then the 2 showing up here is in the plasmid, and you don't see them anywhere else. So because now we have this genomic sequence, so BGI and the Beijing Institute of Microbiology and Epidemiology researchers, we developed a straightforward PCR diagnostic protocol to rapidly identify the outbreak strand. And then this diagnostic method consists of 2 pairs of amplification primers that target the AAFI and STX2 genes. STX2 genes is the Shiga toxin producing gene. And the diagnostic results can be obtained within 2 to 3 hours after receiving the sample, and thus have been extremely useful for detecting this bacteria. So we test the specificity by assessing both computationally and experimentally. Computationally, we compare the primer with 4,547 strands through computational analysis. And then experimentally, we compare with 323 DNA samples. And here is the analysis of computational analysis. And the result is that the kit and the protocol is highly specific. No bacterial strain other than E. coli 0104 had positive implication results of both if they have both targeted regions. Okay, so yeah. So also the diagnostic protocol is very specific and also very sensitive. So it can be as low as 1 picogram DNA is needed for the detection. And then within a few days, we made the primers and also the disease, the protocol freely available to the public. And this is the site that people can download. So I think throughout this process, everything is quite open. So we actually released this data into the public domain, which elicited a burst of crowdsourced, curiosity-driven analysis carried out by bioinformaticians on 4 continents. So about 24 hours after the release of the genome, it has been assembled. And 2 days after its release, it has been assigned to an existing sequence type. And also within a week, about 24 of reports have been filed on an open source wiki, which is a website that facilitates collaborative effort and dedicated to analysis of this strand. So this is really a public effort, and everyone contributed to make the diagnostics quickly available. And also the results now is published in 2 publications. One is in New England Journal of Medicine. The other is a Journal of Clinical Microbiology. Okay. So the conclusion, so we applied multiple sequencing platforms in this study, and we identified the Delhi E. coli as the EAEC strand, which acquires Shiga toxin encoding genes. We identified 8 groups of antibiotic-resistant genes and used the genomic information. We were able to design the diagnostic protocol and primers and made them publicly available. So I guess a lot of you, I don't know if you know, you are familiar with BGI. So I want to quickly introduce who we are. So we are the largest genomic organization in the world today, and we are as a whole is a nonprofit organization. So you see that like we do work like these, like E. coli to benefit mankind advanced science. And we do have commercial services, and all the proceeds from commercial services are applied towards basic research. And then we have established BGI Americas and BGI Europe as interface with both our customers and our research collaborators in Americas and Europe. And today, about 15 of the top 20 global pharmas are our customers. And we have incredible wealth of resources. We have many sequencers, and we have a very large capacity and also the computing power to enable these large-scale projects and collaborative projects and services that we are doing. And we have about 1,000 bioinformaticians developing methods and doing analysis. So we offer a comprehensive service offerings across genomics, transcriptomics, epigenomics, metagenomics, and proteomics. And I think the most relevant one for today is the complete map services for bacterial genome, where we use a number of technologies, including Illumina, Solexa 454, Sanger, Iron Toron, and also optical mapping, and then to assemble gap filling and validation and generate one contig for bacterial genome. And also, metagenomics is a field that we spend a lot of effort on, and metagenomics allows us to explore microbes in their natural habitat, and also this includes both the cultured and unculturable microbes. Well, as you may know, that over 90% of the microbes are unculturable, and metagenomics provide a way for us to study them. So we are involved in the Earth Microbial Project, where we are studying the 200,000 samples together with our collaborators from all over the world, different environment, and also the Gut Microbial Project that we study the microbial and its association with disease. And then in terms of solutions, we offer the whole genome metagenomic sequencing, establish the gene catalog, and also we do the 16S DNA tagging. This is for the taxonomy classification, and we also do metatranscriptomic survey. So here I'm circling a paper that this is we established the first gut microbial gene catalog, and also these are some of the cover publications that we did together with our collaborators. So I guess this concludes my my part of the presentation. If you have need more information, and you can these are our contact for both Americas and Europe.